Good morning, everyone. A warm welcome to all the professors, researchers, teachers, and students who have joined us today on this Gyan workshop from different colleges and universities across Madhya Pradesh and different parts of India. And also, I welcome the Department of Chemistry, IIT Indore, and Department of Bioscience and Biomedical Engineering. Before we proceed, I would like to briefly outline the objectives of this Gyan workshop. This initiative aims to bring international experts to Indian institutions and promote academic collaborations, and also improve the quality and research of Indian institutions and help India to reach globally in science and technology. In line with these objectives, it's our great pleasure to welcome you all in this 10 days Gyan workshop entitled The Exciting Landscapes of Precision Medicines and Next Generation Deep Medicines, sponsored by MHRD, Government of India, and jointly organized by Department of Chemistry, IIT Indore, in association with Purdue University, USA. It's now my privilege and honor to introduce our distinguished resource person for this Gyan workshop, Professor Kavita Shah from Department of Chemistry, Purdue University, USA. Professor Kavita Shah is a distinguished faculty member in the Department of Chemistry at Purdue University. Her work, research, and teaching focuses on chemical biology, cell signaling, and drug discovery, with a particular emphasis in kinase signaling in cancer and Alzheimer's disease. She has made significant contributions to the development of chemical genetic methods for identifying kinase target and published extensively in leading international journals. Earlier, as a group leader at the Genomics Institute of the Novartis Research Foundations, she led teams working on target discovery, validations, high throughput screening, and cancer drug development, leading to the discovery of highly selective and effective kinase inhibitor. At Purdue University, her research group continues to work on outstanding cancer and neurodegenerative diseases with over 100 research publications to her credit. Her work clearly reflects the impactful and meaningful research in modern biochemical sciences. Now, I would like to invite Head of Department of Chemistry, Professor Tushar Kanti Mukherjee sir, to inaugurate the program and share a few words with us.
Yeah, so very good morning to all of you, and you are most welcome and warm welcome to this 10 days Gyan courses organized by Department of Chemistry, Professor Chelvam Venkatesh. And we are truly honored to have Professor Shah with us during this Gyan course, who will be taking various lectures on this precision medicine and next generation deep medicines. So, just to give you a broad overview of our department and the institute. So, Department of Chemistry was initiated in the PhD program since 2009 onwards. So, it is like our 16 years going on, and over the years I think 150 around students have already graduated from our department. These are PhD students, and we also offered 2-year MSc program. Okay. So, and the students are mostly preferred to go abroad for a postdoctoral or doctoral thing and the postdoctoral thing. And from next year onwards, 2016-17, we are having our BS program. So, that is a 4 years BS program, and the intake it will be from the JEE Advanced. Okay. So, if any of your juniors or relatives are there, you please encourage them to register in this program. Apart from Department of Chemistry, we are having 12 departments in our institute, and in this department we also have this various center of excellence as well. Okay. So, and they offer various degrees starting from bachelor's to PhD, okay, MTech, Masters, MS. So, you can go through this program. If any one of you or your relatives, friends wants to enroll in IIT, you should encourage them. And with this, I think if those who are inside, I think many of our students are already here. Those who are coming from outside, you can visit various labs in the Department of Chemistry, interact with the students, so that this gives you a broad overview of the research culture. And we having around 19, exact 19 regular faculty member, and they have their own research lab. So you can go through the profiles if you want to do some internship as well as project, summer project, short-term project. We encourage students actually. So you can directly mail the faculty members, email the faculty members, and do some kind of short-term internship or project. So yeah, so our student volunteers are also there. I am thankful to them as well. If you find any difficulties, you can contact them. And big thanks to Professor Venkatesh for organizing this GAN course. Year after years, he is very active, okay, in this perspective. So anyway, wish you all the best, and I wish grand success of this GAN course. Okay, thank you. Thank you.
Thank you, sir, for your valuable work. So as a token of respect, and I would like to ask Professor Tushar Kanti to welcome Professor Kavita Jha with flowers. Now I request the organizing secretary of the GEARS course, Professor Venkatesh sir, to share a few words about the workshop.
So good morning everybody. Head of the Department of Chemistry, Professor Tushar, and my colleagues Sanjay, Sampak, Selva Kumar, and also Professor Meena Tiwari from SGS ITS, students from DAVB, SGS ITS, and other universities across India, also from the Department of Chemistry and Department of Bioscience and Biomedical Engineering. So I welcome you for this wonderful event, and I believe that you will enjoy for the next 6 days one of the very important topic in healthcare. So cancer, diabetes, and neurodegenerative diseases, and what are the root causes and how drugs were discovered in the world. And this will be enlightened by the visiting faculty from Purdue University, Professor Kavita Jha. And our association with Professor Kavita Jha goes to more than 2 decades. So Professor Kavita Jha was an alumni of IIT Kanpur, and she was an excellent organic chemist, maybe senior to me 10 years before I graduated from IIT Kanpur. And once she moved to United States of America, so she became a chemical biologist, an organic chemist turning into a chemical biologist, a full-fledged chemical biologist, having complete knowledge of chemistry as well as biology. She was working in Novartis and developed several kinase inhibitors, because of which she got an invitation from Purdue University to join as a faculty in the Department of Chemistry without tenure track. She was appointed as an associate professor and then moved in the ladder to become professor in the Department of Chemistry. So destiny has a different destination. So when I was doing postdoc at Purdue University with Professor Phil Lau, so we were working on animal studies for imaging and therapeutics. So during this time, Madam was involved in developing kinase inhibitor, especially for understanding the growth of cancer. So one day she approached Professor Phil Lau and requested that she want to do some animal studies, implantation of the tumor, because she has developed some cancer cell lines by knocking out genes, whether these genes are responsible for the cancer growth or not. And this is how I met her for the first time, and we did animal studies implanting tumor and the tumor ablation, and it worked out fantastically. And the concept of knocking out aurora kinase or kinase inhibitor proved to be the right. So this was the first time I associated with her, and then within 6 months I moved to IIT Indore as a faculty here. So later, our students visited from bioscience and biomedical engineering for a year to do research in her lab. And further, Leknath also visited recently to United States. So wherein the molecules we developed at IIT Indore was found to have very precise kinase inhibition of various kinases. So which is currently under publication, and we also patented it recently. And further, Madam visited in 2019 before the COVID set in 2020 for a month as a overseas visiting doctoral faculty fellowship at IIT Indore. So really it is a great pleasure, and you will see the depth of the knowledge Madam has about cancer and diabetes and neurodegenerative diseases. So I would welcome Madam to IIT Indore and also to the Department of Chemistry, and I hope you will enjoy her lectures. So for the coming next days, like she said before, that you can interrupt her anytime and inquire her if you have any doubts, and feel free to. Thank you so much. Thank you.
Thank you, sir, for your kind words. Now we will begin our first lecture of our Gyan workshop. So I would like to invite Professor Kavita sir to deliver his first talk. Thank you, ma'am.
Okay. First of all, I would like to thank you all for inviting me for this prestigious Gyan course. And it is so nice to meet so many old friends. I have been here twice and I know many of you, so it's a real pleasure to be here. And Venkatesh is a pioneer in organizing Gyan course year after year after year. So this is my second Gyan course, so thank you so much for inviting me once again. So, all right, let's get started then. Okay. So as I said, please interrupt me at any time if things are not clear. I want this to be a very interactive session. Okay. So, I will start with the introduction of cancer. So this is a brief outline about the things. You guys can hear me, right, at the back? Okay, good. So this is a brief outline of the topics that I'm going to discuss today. So I will start with first 14 hallmarks of cancer. So when you hear about cancer, and if you have to explain cancer to a layman, what is the thing you say? What is cancer? Cancer is just uncontrolled cell growth, right? That's what cancer is. That's how scientists used to define it. The second hallmark of cancer is that it metastasizes. It goes and lodges somewhere else. So these were the two major hallmarks of cancer. That's how it started. And then Bob Reinberg, he then said, oh, it has many more features. So he made that figure which I am going to show you, and it had 10 features of cancer, the 10 things. And very recently 4 were added. So now there are 14 hallmarks of cancer. So we'll briefly look at those today. And then a number of topics which are linked to cancer. So let's start with these hallmarks of cancer. Okay. So, so this is a tumor, right? Cancer is a tumor. So one of the hallmark of cancer is that it grows uncontrollably. That's the major hallmark. It keeps growing, 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 no control. So all the cells, they get signal, and when they get signal, they divide, and then they also get a signal, stop now, and they stop growing. So in cancer, there is no stop signal, they just keep on growing. So that is the major thing. And so, it is very important for cells to die when they are supposed to die, okay? But cancer cells don't die, they resist death. So what happens, there is always a balance there. Cells are growing, cells are dying, and this is the correct balance. But in cancer, cells are growing like this, and they are not also dying. So they keep on increasing in number, okay? So they escape cell death, okay? Now you can imagine that suppose this is a tissue, right? So this is a tissue and cancer is growing like this. So the tumor is coming like this, okay? Now usually all tissues, all cells have blood supply, and this blood supply gives oxygen, right? Because they need oxygen. We know Krebs cycle requires oxygen. It gives all the nutrients here, okay? But when the tumors are growing, now as they become bigger and bigger, these tissues, they don't have blood supply. They don't get nutrients, they don't get oxygen. So what these tumors do, they promote angiogenesis. And what is angiogenesis? Angiogenesis is growth of blood vessels. Okay, so that is a hallmark of cancer. So suppose this is a tumor, so now blood vessels will come and surround it, and these blood vessels will bring both oxygen, nutrients, everything. Okay, so if you inhibit angiogenesis, you cannot cure cancer, but you can stop it for a while, but after some time they will resist that and they will find another way. Okay, so this is another hallmark of cancer, which is angiogenesis. Okay, so this is because of angiogenesis, they have increased nutrient and oxygen supplies to the tumor. Okay, now this is one hallmark. This is that initially, if you look at old textbook, people used to say cancer is a genetic disease. It is not only a genetic disease. Actually, usually 5 to 10% is due to defect in genetics, but the rest is sporadic. Okay, so if we smoke, we are creating cancer in our body. If we go out in sunlight for very, very, very long period without any protection, we are inviting cancer. It is not genetic. We cannot blame our genetics. So genetic contributes to cancer. Some of you may have heard of that in some family, women are very prone to breast cancer, okay, because they have BRCA1, BRCA2 mutations. We are going to discuss that later in this course. So they are prone to breast cancer. I have known a family where every female in that family has breast cancer. So there it is the genetics, okay? But fortunately, it's only 5 to 10%. 95% or 90% is sporadic, okay? All right? So genetic can contribute, but the biggest one is that they spread to other part of the body. Cancer, unfortunately, is rarely localized. Okay, it starts with a localized cancer, but then it metastasizes, and which is very bad. Okay, so this is a recent one, but Bob Reinberg was the one who initially showed this. This is one of the most famous figure of cancer, where he showed what are the features of cancer. So very briefly, so first one is sustained proliferative signaling. Divide, 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 divide. Then at the same time, anything which resists growth, evade them. Okay, so they don't take no for an answer. They keep on dividing. Okay, they resist cell death. And then so what happens is that if you go out in sunlight for very long, okay, if you are exposed to UV light or X-ray light, or any kind of chemicals, okay, bad carcinogenic chemicals, you introduce mutations in your body, and those mutations can lead to cancer. And that is what is meant here by genomic instability. If they are mutation, they can lead to cancer, but we are inviting them by going out in UV light for a long time. Increased vasculature, angiogenesis, metastasis. Now, many times people have accidents, right? And the body is inflamed, and the doctors often give you something to reduce inflammation, right? When they give you, so many people, what they say, oh, I will bear the pain, I don't want to take these many medicine, but it is a very bad idea to have inflammation in your body for very long. If you have chronic inflammation, it can give rise to cancer. Okay, so I have known some people who had accidents and they were given a lot of medicine, and medicines were also making them sick. So they decided, I'll take the pain. Okay, no, you have to reduce the inflammation. Inflammation is very bad. Inflammation can give rise to cancer, it can give rise to neurodegeneration, it can really create problem. So you have to reduce inflammation. One of the thing, right, we all eat haldi in our food, right? Haldi reduces inflammation. Okay, that is why it is so good. Okay, so inflammation, so this inflammation is really bad. Now this part here, avoiding immune destruction. How many of you have heard of immunotherapy for cancer? Anyone? You have. Okay, that is the golden thing for cancer these days. It's a game changer for cancer, immunotherapy. So what cancer does, that you know, you will be surprised to know in our body every single day, hundreds of thousands of reactions are going in our body every day of our life, which can lead to cancer. But we don't get cancer because the immune system fights it off. Okay, so our immune system is taking care of all the wrong things which are happening in our body every single day. Okay, so what cancer does, it fools the immune system. Okay, and that is something we'll discuss in detail. So cancer will say, oh, I'm yours. So immune system does not recognize it as a foreign thing, and it shuts down the immune system. So the most recent drugs are immunotherapy, and these immunotherapies have saved many, 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 many patients. It is a very powerful thing. However, you know, cancer always, I mean not always, in most of the cases it will resist everything at the end. But immunotherapy is still extremely good. So cancer cells avoid immune destruction. So these were the initial 10 features, but very recently these 4 were added. Everything we'll discuss in detail eventually, but I want to tell you what these 4 things mean. So I'll start with polymorphic microbiomes, right? So we all know that we have beneficial bacteria in our gut, right? We all know that, right? They help in digestion. But what you might not know, that this microbiome, so this microbiome is present in all over our body, in our saliva, in our nose, all over the body. And if your microbiome is healthy, it will resist cancer. Okay, these people are not going to get cancer. But if your microbiome is not healthy, it can promote cancer. Okay, so this is polymorphic microbiome. And then this is very interesting. Okay, so there is this really aggressive form of prostate cancer, very, very aggressive form of prostate cancer. So what happens when people get prostate cancer? The first step is surgery. Okay, so you can remove it by surgery, and that is called castration. So you remove it. Many times people survive for many, many, many years and it never comes back. But in most of the people, it comes back after some years, and it is called castration resistant prostate cancer, or CRPC. Okay, so then there are many drugs for CRPC also, right? So initially when those drugs were discovered, scientists thought, oh, we have won the game with prostate cancer at least. These drugs are so effective. But then after these drugs were given to the patient, a new form of cancer emerged which never existed before. This is man-made cancer, because now the cancer cells developed this new thing. So now, you know, what is cancer cells? They are cancer, like they are undifferentiated on top of each other, right? But now they become neuron-like, completely neuron-like. Have you ever heard of cancer being neuron-like? You have seen neuron pictures, right? They are neuron-like. So they completely change themselves. They don't look like anything like before. They don't, if you keep on adding cancer drug, nothing happens because they are neuron-like. Okay, these are called NEPC, neuroendocrine prostate cancer. That comes under phenotypic plasticity. So suppose I am Kavita Shah and I become someone completely different person who has completely different identity. That's what they do. So now the old drugs have no effect on them because they have changed their appearance, they have changed their pathway, everything. their survival depends on something else. So you can keep on adding the same drug, they are resistant. So that is phenotypic plasticity. They just change their appearance, pathways, everything. Okay. Then how many of you have heard of epigenetics? Anyone? You have. So most of you have not heard of epigenetics. So we know mutation, right? Change in DNA. That you all know, right? So there is another thing called epigenetics. So what happens is that in this DNA sequence doesn't change at all. Okay, but other things are added close to DNA. Have you ever heard of DNA methylation, histone modification? That's epigenetics. So what is the significance of that? So just to give you a little bit of background, human body has 6 billion bases, 3 billion base pairs, all of us, okay? And you will be surprised to know that all the human beings in the entire world, we are at least 99% identical, maybe 99.5% identical. So we have all different appearances all over the world, right? Everything, we appear very different from each other, but we are 99.5% identical, okay? So coming back to epigenetics, so we have 6 billion base pairs, but a very, very, very small percentage of that is expressed as proteins. So you know DNA makes RNA, RNA makes protein. So 5% of that 6 billion bases are expressed as protein, right? So 5% of the total human DNA makes proteins. Makes sense? Makes messenger RNA, which are responsible for forming the proteins. So what determines that 5% there? So every cell of our body, every single cell has exactly the same DNA. From the head to toe, every cell has the same DNA. But you all know neurons look very different than our skin cells. They have very different functions than our skin cell. Our intestine cells have different functions, right? Our stomach cells have to release HCl. Our liver cells have to do something else. So if the DNA is identical, why they are so different? Why do they have such different functions? Because they are expressing different proteins. So if this DNA is identical, why they are expressing different proteins? Because they get the signal, you have to express these proteins, and these neurons have to express these proteins, and that is called gene expression, and that is determined by epigenetics. So what happens is that, I will tell you in details later on, because this is just an introduction, but epigenetics tells you these genes will be expressed in the brain, these genes will be expressed in the skin, and that is why they all have unique functions. They're always housekeeping genes which are expressed in every cell of our body. Okay, you know glycolysis, right? Krebs cycle happens everywhere. So those proteins are present throughout the body, but then these guys have unique proteins, these have unique proteins, these have unique proteins, liver has unique proteins. That's gene expression, and that is controlled by epigenetics because DNA is exactly the same. Okay. So there is no wonder cancer cells change the epigenetic programming also. Okay, so that is what is meant by this, and I will come to senescent cells later on. Okay, so this is all those things. So I am showing you many, many pictures which have a lot of information. So you don't have to pay attention to every detail. Don't pay attention to all these signaling pathways. It's part of the picture. You don't have to pay attention to that. Just pay attention to these things here. Okay, the blue ones. What are the properties of cancer? Okay. So I will briefly go over these things. So first is sustained proliferative signal. So the way it happens in normal cells, that normal cells get a signal from outside, growth factor, and there is a receptor. So this receptor, when it binds to the growth factor, signal is transmitted to the nucleus to tell, okay, you divide, okay? And then this growth factor will leave, and once it leaves, the signal disappears, and the cells will stop dividing. Is it making sense to you? So they will get the signal and then the cells will divide, and then the growth factor will go away and it will stop. If the growth factor doesn't go away, we all have experience, right? When you're cooking food, you can smell the food, right? But if you stay in the kitchen for very long, you stop smelling anything, right? And then you go away and you come back after some time, you can smell again. So why can't we smell after some time? Because the receptors, they are internalized. Signaling is turned off. Now we cannot smell anything. And now we have to give it time so that they can come back up again and we can smell again. So in under normal condition, every signal has to stop after a while. Okay, that is how it works. So in cancer cells, there are many, many mechanisms. But what happens is that proliferation signal is going all the time. Sometimes it becomes independent of growth factor. It is just on all the time. Okay, and the cells keep on dividing. Okay, so that is that one. Okay, second thing is that every cell has tumor suppressor proteins. These are called growth suppressor. So they keep the proteins, cells growth under control. How many of you have heard of the protein called P53? Yeah, you all have heard, right? P53 is a tumor suppressor. Why it is so famous? When P53 was discovered, it was discovered by a scientist named Arnold Levine, who was at Princeton at that time. So when P53 was discovered, he used to tell us a story that he thought it was actually a tumor promoter. And he thought that this is it. P53 discovery will eradicate cancer forever. That didn't happen because it turned out to be a tumor suppressor, but probably the most famous tumor suppressor. So what cancer cells do? They inactivate P53 or make P53 disappear, and cancer just keeps on growing. If you put back P53, cancer will come under control again. Okay, so they evade growth suppressor. They resist cell death. Anytime cell death signal comes, they resist. Okay, so then how many of you do cell culture here? Yeah, many of you do cell culture, right? So when we do cell culture in the lab, the cells are dividing all the time, right? Infinitely they can divide because they are immortalized. But if we take our skin cells, it will probably divide for 2-3 generations and then they will die, right? So all these cells are supposed to die after a few generations, but cancer cells are immortalized. They just keep on dividing. They never die, right? Okay, which is very good in the lab, but in human body, they need to die. Okay, okay. Angiogenesis, like I said, is a growth of blood vessels. Okay, so this is another story. So the person who discovered that angiogenesis occurs in cancer, so he said, okay, give angiogenesis inhibitor. I have seen all of these discoveries in my lifetime. Okay. So angiogenesis inhibitor, it became such a huge story. We thought we have won cancer battle. You give angiogenesis inhibitors, blood vessel growth will be inhibited, cancer cell will be starved, no food, no oxygen, they will die. It is effective initially, then they can learn to survive even in the absence of oxygen. Okay, and they use it for their advantage. Okay, so the bottom line is that for cancer, you should not give, the therapy should not go very long, because if it goes for very long, they develop resistance mechanism. You have to go there and use a shotgun and kill it very quickly before, like, without touching the normal cells. That's this course is all about, how you can kill cancer without touching the normal cells. Okay, all right, so this is angiogenesis. So this is a blood vessel and keeps on growing. So these are tumor cells. So initially they don't have blood vessels, they are low in oxygen, low in food, and then they grow tumor like this. Okay, they grow the blood vessels. Now they are very happy. Tons of cancer, by the way, loves sugar, really loves sugar. So more sugar, they are super happy. Okay, so so this is what is angiogenesis. Genomic instability. So we all know that if we are exposed to X-ray, when you go to X-ray, they protect your all other organs, right? X-ray is very bad. So any kind of radiation can lead to DNA damage, and if DNA damage is not repaired, that will lead to genomic instability, and that is a sure way to get cancer. Okay, so it is actually, I know of a case, there was a person, she had thyroid issues, right? So she told me that she's getting her thyroid out, okay? So what they do is that they irradiate the thyroid. Many people take levothyroxine, hypothyroid or hyperthyroid. It's a very common disease, right, worldwide. So she had one of those, I don't remember. So they told her that they will irradiate her thyroid, take it out. She had no cancer, nothing, perfectly healthy lady. So, and they irradiated her, and they irradiated her too much, and then she developed the cancer, and she died within 6 months. So it was doctor given cancer due to irradiation. Okay. So radiation is really bad. If you work with radioactivity, you know how much precautions we take if you work with radioactivity, right? Because it damages the DNA. DNA damage, if not repaired, can lead to cancer. Okay. Inflammation, like I said, is very bad. If there is inflammation, you have to take anti-inflammation medicines to keep it down and bring it down and keep it down all the time. Okay. Do you guys know how much curcumin is a component of haldi? You know that, right? You know that it is like there are, I don't know how many pharmaceutical companies who are trying to make analogs of curcumin which are more bioavailable. So in its current form, if you eat in curry form, our body absorbs some percentage, which is very good. But if you take a capsule of curcumin, it is ineffective. It does not help at all. So although it's very common that people who do not eat curries, they take capsules of curcumin, turmeric powder, which is useless. So they are trying to try to make analogs of curcumin so that it is more bioavailable. But the point here is why it is so good. One of the property of curcumin is that it reduces inflammation. Okay, it has many other properties, but this is one of it. Okay, this is the worst part of cancer, that it metastasizes to other parts of the body. So here, this is the primary tumor. Now it is slowly invading. I will show you many things, and you will understand exactly how it invades. So here it is invading. Then it will break open the blood vessel. Once it comes to the blood vessel, blood goes everywhere in the body. So now it has access to the whole body. It will find a spot and it will grow there. Okay, so this is the worst part. And once this metastasizes, then it is pretty bad situation. Okay, so this is the metastasis. It has broken this boundary. Now it is going to the bloodstream, and with bloodstream, it has access to the entire body. Okay. In this case, it is metastasizing to lungs. Okay. Then avoiding immune escape. So our immune system is working every second. Okay, and it is keeping us safe. So even if you don't go out in sunlight, our DNA gets damaged spontaneously. I don't know, maybe 10,000 times a day. Okay, and it is getting repaired all the time also. But if DNA is damaged, and if then the cells are dividing uncontrollably, the immune system kicks in and it finds it, it cleans it up. So immune system. So any time the cells are bad, immune surveillance picks it up, eliminates it. We are safe. But what happens, cancer cells, as they grow, it can become a cancer if the immune system is not working. So what they do, they act and they fool the immune system. And immune system thinks, oh, it is self, it is the right thing. They will form, this is an immune cell, and they will make bonds with the immune cells. Immune cells thinks, oh, this is the normal way. So immune system shuts down, and that's what leads to cancer. It's a major cause of cancer. So if you give immunotherapy, you reactivate the immune system. Okay? So, and then I have seen the rise of this field. So I used to teach metabolism to my students, and metabolism had nothing to do with cancer. And so Louis Cantley at Harvard, he published the first paper in 2004. That time I was teaching metabolism to my students, and he was the first one to show that cancer cells have altered metabolism. And now there are maybe 10,000 papers on cancer and metabolism. So what cancer cells do, they change our metabolism. Okay, like I said, they love sugar. Okay, so they really love sugar, and they make, you know, everything which is bad, they make it in such a way that it serves them. It serves cancer. So what, in a very simple words, what happens is that normal cells will take glucose, glutamine, lipids, all of these things. But this is proliferating cell. So this will take a lot more glucose, a lot more glutamine, not as much lipids. Why? So, you know, you guys know what is glycolysis, right? All of you. You all know Krebs cycle, right? So what is the advantage of Krebs cycle? Krebs cycle makes ATP, right? Krebs cycle is not making ATP, but it is making NADH and FADH2, which feeds an electron transport chain, and it gives you huge amount of ATP. So as scientists, if I ask you, cancer cells should have higher glycolysis, higher Krebs cycle, does it make sense to you? So glycolysis is the one which takes glucose, converts it to pyruvate. Then pyruvate is converted. I know you guys have very diverse background. Some of you may know, some of you may not know. If you do not know, please tell me. I will write it down here. Do you have chalk or something?
Yes.
Okay. That's okay. I don't need it right now, but from tomorrow onwards, we'll have chalk here so that I can show it to you. So in case you don't know glycolysis, in glycolysis what happens, you start with glucose, and in 10 steps you change glucose to pyruvate. Okay. Then in one step you change pyruvate to acetyl-CoA, and that is called transition reaction. Acetyl-CoA starts the citric acid cycle. Okay, citric acid cycle. Citric acid cycle will produce NADH and FADH2, which then feeds into electron transport chain to make tons and tons of ATP. So now I ask you, in your opinion, and cancer cells are dividing all the time, and you know you need energy if you want to divide and you want to do. So according to you, should these cells have higher glycolysis and higher citric acid cycle? Yes, right? It makes sense. And that is true. One of the way to identify cancer inside the body is that they give you radioactive glucose, and then they do PET scanning, and they can pick up any PET scanning. They will see, okay, there is the brain cancer or lung cancer. If there is any dot anywhere, they will pick it up. That's how they scan the whole body to see metastasis, right? Because cancer cells love glucose, and there is huge amount of glucose consumption where cancer is present. So that is the way, that's a diagnostic tool for cancer, okay? So do you think that cancer cells only require ATP or something else? If I keep on feeding ATP directly to cells, will that be enough for cancer cells to form the tumor? Just say yes or no. Raise your hand if you think it is yes. Raise your hand if you think it is no. Correct. It's a no. Why? Just ATP is not enough, because they have to build another cell, so they need all the lipids. If you have to you have to make another room, you need everything for this room. You need the chairs, you need the bricks, you need every single thing. So if you're duplicating a cell, you need all the things. So they need lipids, fats, everything, right? So what they do, they eat a lot of glucose. They make ATP, but they also take the intermediates of glycolysis, and they send it to build all the other macromolecules to build the cell. Okay, so that is why it makes sense that the metabolism is also different between normal cells and cancer cells. Normal cells will have fast glycolysis, cancer cells will have slow glycolysis, because all of these intermediates go in different directions to build the whole cell. Okay, you need starting materials to build macromolecules. Okay, all right, so this is what it is showing, that it has completely different cell metabolism. Okay, so this is again showing you that. So like I said, in many of the slides you have too much information. You don't have to memorize all of this thing. Get the gist out of it. Okay, that the metabolism is different between normal cells. So all of these metabolisms are different in tumor cells. Okay, so now the next part. How do you classify cancer? Okay, and there are many ways of classifying cancer, many ways of classifying cancer. How I'm doing with time? My watch is not charged, so that's why half an hour, plenty of time. Okay, so there are many ways to characterize a cancer. What is the simplest way to characterize a cancer? Lung cancer by tissue, lung cancer, prostate cancer, breast cancer, brain cancer, blood cancer, like this, right? We all know this thing. But there are many other ways to classify cancer too, and every type of classification is very important. Okay, so you can classify cancer by site of origin, which tissue, right? Which is the common way we say, okay, this is this cancer. Then the other one is by tissue type. So whether it is you guys know ectoderm, mesoderm, endoderm, right? Blood, all these things, different tissue type. So depending on which kind of tissue they are originating from, we call it like carcinoma, sarcoma, myeloma, leukemia, lymphoma. So I'll tell you what are these. Carcinoma arise from something which is the outermost membrane, outermost layer of cells. So skin is obviously outermost, but if it is liver's outermost layer, that is also outermost layer. So have you heard of liver cancer? I mean, obviously you have heard of liver cancer, right? So liver cancer usually is HCC, hepatocellular carcinoma. Okay, carcinoma means it is the outermost cell of the liver. So all the outermost thing of any organ, that is carcinoma. Okay, sarcoma begins in bone or in soft tissues of the body such as fat or muscles. Okay, myeloma becomes in plasma cells or white blood cells. I have these in details also later on. Okay, leukemia, we know this occurs in bone marrow of this, and bone marrow makes red blood cells, white blood cells, so that cancer is pretty bad. Lymphoma is in the cells, lymph nodes, lymphatic system. That's very bad because lymph goes throughout the body, so it spreads very fast. Okay, then it is by grade and by stage, by spread. So let's look at each of this. These are the 4 kinds. So actually by stage and grade, and then there is a molecular classification also. Please ask me if you guys have any question. So far, any question? Let me pause for a second. I'm sure something did not make sense, right? Up until this point. So ask me. Yes, please. Thank you for asking the first question.
One of the factors you said, sugar.
Sugar, yes.
They like sugar.
Yeah.
Now I'm seeing studies that they say that if you want to counter cancer, make your body alkaline, avoid use of acids. So what's the rationale?
So, so I will answer it in 2 different ways. When it says sugar, why they call sugar? Because glucose, they eat glucose. And why they eat glucose is that, so I did not tell the whole story in interest of time. So what Louis Cantley showed, so everyone in the world used to think that glycolysis is fast in cancer cells, because the faster it will go to pyruvate, faster it will go to Krebs cycle and more ATP will be produced. So what Louis Cantley showed that glycolysis is slowed down in cancer. And when it is slowed down in cancer, what is the advantage? These intermediates can escape in different directions to form cancer cells. So the whole bottom line is that glucose is eaten by cancer cells in huge amount. But not everything is converted to pyruvate, right? So that is the sugar rationale. The second thing is that I did not go in details also glutamine. So glutamine is also utilized by cancer cells in huge amount. Cancer cells are addicted to glucose and glutamine. Now when they say about the alkaline diet that you are asking, it affects cellular metabolism. Okay, so acidity is very bad, acidity, because when you have acidic environment, cancer cells thrive in acidic environment. So when I briefly said, things which are disadvantageous to us, cancer cells actually use it for their advantage. So what happens in acidic environment? So cancer cells produce lactic acid. Okay, that lactic acid is in the tumor environment. When it is very acidic, when immune cells come to it, immune cells become inactive in the acidic environment. The acidic environment helps in breaching all this basement membrane. Cancer spreads around it. So then acidic environment changes cellular metabolism. And when it changes cellular metabolism, it helps cancer cells. It does not help the normal cells. So when they say you have to keep your body alkaline, it helps the whole homeostasis of the body. But keeping it alkaline alone is not going to make an impact on cancer. But having said that, something which really helps in preventing cancer is what we eat. That really helps in preventing cancer. So I don't know, I honestly don't know the percentage of cancer in India versus in Western countries. I don't know, I have not studied that. But what my understanding is about brain also, Alzheimer's disease, that is for sure that people in Western countries, they get a lot more Alzheimer's than people in India. It could be because people in Western countries, they live much longer life. I don't know why that is the case, but they have much longer life there than here. And Alzheimer is an age-related disease. The longer you live, the more chances that person has. But even if we live to 80 or 90 or even more than 90, we don't get Alzheimer's because of our food thing, because we have very high turmeric powder in our food thing. So food definitely helps that. So alkalinity in the body really helps to prevent a lot of diseases. It keeps our homeostasis in balance. Acidity is pretty bad for our body. Any other question? Yes, thank you. Go ahead.
You said that cancer cells have a slow glycolysis process. So
Yeah.
that would imply that it has a slower rate of ATP production as well.
That's a very good question. Okay. So what happens is that, so that is why when this study was discovered, so everybody in the world thought the cancer has to be, glycolysis has to be much faster because they need more ATP, right? But this guy showed that no, they have slower glycolysis. This was against every study which was done before, right? And because it was against the flow of the knowledge at that time, this came out in Nature. And when this paper was published, that guy said, the paper says very clearly that this is slow, and they showed definite proof that it was slow, but they could not explain why it was slow, because it was like nobody would believe it, but they showed it conclusively. Then 2 months later, 3 months later, they published another Nature paper, and there they showed why it was so slow. And the reason is this. So those of you who have studied glycolysis, so it starts with glucose. Glucose is converted to glucose 6-phosphate to fructose 6-phosphate to fructose 1,6-bisphosphate. then it gets cleaved to 3,3 sugar, right? Glyceraldehyde 3-phosphate and goes on, right? So what happens is that, in interest of time, I will not tell you all the things, but if it is going fast like this, right? So those of you who have studied glycolysis, there are 3 steps which are irreversible. All other steps are reversible. So these 3 steps are the one which decide how fast the reaction is going to go. So what he showed, the last step, which is catalyzed by pyruvate kinase, is slowed down in cancer. So suppose if I keep you busy and I'm the first step and we have to transfer things out of that door, right? And there are 10 of us transferring things. And if the 3 people who are keeping it flow going, things will keep on going out, right? But if people slow down, what are you going to do? We are going to start talking to each other and this. So when things slow down, these intermediates, they move out because things are slowing down. So glucose is still consumed a lot more, but the output is less here in terms of how much glucose is used, because these intermediates are going in other directions, and these intermediates serve to make other macromolecules to make a cell, because ATP alone cannot make a cell. You need every single thing to make a cell. And so let me put it in a different way. If normal cells are using 100 molecules of glucose per, it is so weird, like it is probably 100,000 per minute, but I'm just making it 100. Normal cells will use 10,000 molecules. If this is 100, they will use 10,000 molecules. So they are still making more pyruvate than this guy because they're using 10,000, but they are not making it 10,000, 100 to 10,000. So it's 1,000 times more, right? So they're not making 1,000 times more pyruvate. They are still making a lot more pyruvate than normal cells, but they're also supplying in other directions. Is it making sense? All right. So very good question. Go ahead.
But doesn't that induce that feedback inhibition as well?
That's what the paper said. So our cells, hopefully in all our bodies, pyruvate kinase is inhibited by, can you tell me what? Fructose 1,6-bisphosphate is the inhibitor of pyruvate kinase. That feedback inhibition is broken in cancer cells. That's why, let me correct myself. Yeah. So fructose 1,6-bisphosphate inhibition occurs in cancer cells, not in normal cells. So in our body, that inhibition is not occurring. So pyruvate kinase is very quick, okay, and it is getting to pyruvate. In cancer cells, that inhibition is occurring. Pyruvate kinase slows down, intermediates move in other direction. Is it making sense? Yes.
Well, glucose and glutamine intake is high in cancer cells, but that is not for lipids. So I want to know whether how the role of
lipids are still taken by cancer cells. Lipids are still taken up, but not as much. So I can show you why glutamine is taken so much, because it forms a lot of things which are required by cancer cells. So lipids are still going in, but glutamine and glucose a lot more. That's what it meant there. Any other question? All right, let's go back to classification of cancer then. Okay, so this is by tissue of origin. So like I said, if they are coming out from the outer layer, right, whether it's liver outer layer or skin or any other outer layer, that is called carcinoma. So liver cancer, most common one is hepatocellular carcinoma. That means it is coming out of the outer cell of this. Sarcoma is muscle, fat, bones. Those kind of cancers are called sarcoma. Myeloma is a cancer that originates in blood plasma of the bone marrow. Okay, leukemia is pretty bad. Okay, it's the cancer of bone marrow and white blood cells. Okay, usually we call it blood cancer. Okay, Lymphoma develops in the glands or nodes of the lymphatic system. Lymphomas are usually extremely bad because it spreads very quickly. And then they are mixed type, which have more than one tissue type. Okay, so this is the same thing what I told you, different kinds of, and it is based on the tissue of origin. Okay, then this is very simple. By primary site, lung cancer, breast cancer, liver cancer, prostate cancer, brain cancer, okay, skin cancer. Then this is another one. By stage and grade. I should not ask this question, but I still will ask you. Have you ever heard of grade and stage of cancer? Of someone that you knew? You have, right? So this is what is stage and grade. This tells you how aggressive the cancer is. So this is obviously very important information. So this is the first thing pathologists will tell you, whether the cancer is contained or spread out and how bad it is. That's what it is telling you. Okay. So what happens is that if you look at this thing, so normal cells look very organized. So if you look at the wall, all the bricks are nicely organized. I mean, obviously you don't see bricks here, but if you just see the bricks in any wall, it's properly arranged. So normal cells, they look like this. When they go and they start to become cancerous, they change appearance. They are no longer properly arranged. They are going on top of each other. Here this is even worse there. So this is well differentiated, and then they don't have time to differentiate. They will start accumulating on top of each other. So this is second grade, this is grade 3, and it becomes very bad. Okay, so these are undifferentiated cells. So less differentiated, higher grade, worse cancer. If it is differentiated, it's still early stage of cancer. So that's what is meant by grade. So grade 1 tumor cell looks like healthy tissues. Okay, grade 2, they are abnormal, they start to accumulate. Grade 3, even worse. And grade 4 is very, very bad cancer. They are just dumping on each other. No differentiation. Okay, so that's what, this is another picture of these grade things. So well differentiated. See, look at the shape of cancer, these normal cells. And now they are just dividing. Okay. So then comes the stage. So stage is given by T and M. Okay. So T stands for tumor, N stands for nodes, M stands for metastasis. Metastasis is always bad. Okay. So tumor, this component describes the size and the extent of the primary tumor. So you know the meaning of primary tumor. So suppose liver cancer is occurring. So the first tumor will occur in the liver. That's the primary tumor. Then liver cancer metastasizes to lungs. So lung cancer will be secondary tumors. Wherever it metastasizes, those are secondary organs. Then it goes to the lymph nodes. And we will spend some time on lymph nodes. So there are about, all of us, we have about 600 lymph nodes. While lymph nodes are important for us, they are part of the immune system. Okay, so once it goes to the lymph nodes, lymph is going throughout the body, so it spreads very quickly. So once it goes to the lymph nodes, it's considered really bad cancer. Okay, so tumor, nodes, and metastasis means you actually have evidence that it has metastasized in the lungs. Some cancer, they metastasize to the bones. Some, they go to the lungs. Okay. Metastasis is obviously very bad. Okay. So this is how stage 0 shows. So now we are in the TNM scale. So it is 0, 1, 2, 3, 4. 0 is good, 4 is very bad. Okay. So 0 indicates carcinoma in situ. Everything is 0. N is 0, M is 0. And in situ means it is contained. Wherever it is contained in that organ there. Okay. Then stage 1 still is localized tumor, and if it is localized tumor, you can remove it by surgery because it is still localized. But we know that cancer patients are given chemotherapy. Why they are given chemotherapy? Because chemotherapy goes throughout the body. It's systemic therapy. That means cancer has probably spread to other parts of the body. So that's why, and that is very toxic. Okay. Then stage 2, that it is locally advanced cancer, T1, T2. T1 is again telling you the tumor size. N1, it is now going to the nodes, which is bad. But it has not metastasized as yet. Then T1 to T4, tumor has gone really big. N2, more nodes are involved. Metastasis, no good news. And at the end, it has also metastasized. So that's very bad news. Okay, so this is TNM staging. Okay, so when it is TX, you are not really sure whether the person has cancer or not cancer. Then T0, probably not cancer. Then you know it is carcinoma, but it is very contained. And then it is slowly penetrating close, and then it becomes T1. It keeps on going. Same thing, no node involvement, no lymph node involvement, and now it has gone to lymph nodes, and now it has metastasized. Okay, so higher number, very bad. Okay, so now we come to molecular classification. So whatever I told you about this thing, this is applicable to all kinds of cancer. But now we'll say, okay, let's go and see what is unique about this particular cancer. Okay, so have you guys ever heard of HER2 positive breast cancer, HER2 positive lung cancer or HER2 positive cancer? You guys have heard of it. What is HER2? HER2 is a receptor. And if that is present, that is molecular classification, because you are telling about a protein in the cell. Oh, this is highly expressed. That is molecular classification, right? It's not a stage, it is not tumor size, it is nothing. Now you are telling specifically about that cancer. Like Venkatesh was saying, that when we collaborated together, right? So that time, our lab did not do animal studies. So with collaboration with Venkatesh, first time, that was our first experiment in the animal models. He taught our students how to do animal model. He was an expert in animal model at that time. He is, but he taught us, right? So that time we discovered that if you remove this molecular this protein called LIMK2, cancer disappears completely. So that was a molecular classification. We identified the culprit in that cancer. So if you can tell the culprit in a cancer at a molecular level, this protein, this RNA, this is responsible, that's molecular classification. And the more precise you are, the more precise medicine you will give to kill that guy there, right? So that's molecular classification. So that will tell you exactly. Usually it is not a one thing. Usually there are many things which go wrong in one cancer. But if you can identify those real drivers, that is the best way to treat a cancer, okay? So molecular classification includes genetic mutations. Like I said, that breast cancer is very prevalent in some families. Almost all females get breast cancer because they have genetic mutations. Okay, so they are very prone, like BRCA1, BRCA2 mutations can lead to breast cancer. You guys all have heard of Angelina Jolie, right? The actress. So she had this surgery to get her ovaries and breast removed. Why? Because in her family, they are very prone to have breast cancer and ovarian cancer. So she got genetic testing done. She had the same mutation, BRCA1, BRCA2, and then she proactively got it removed so that she doesn't get this cancer. Okay, so it's becoming a very common thing now. People get it tested if they have a family history, and you can proactively, get rid of that organ or do something proactively so that that person doesn't get the cancer. Okay, so that is what it is. Then somebody gets the cancer, you do gene expression. So like HER2 positive cancer, triple negative subtypes. So in Western countries, people in US, people who are African Americans, so who originally were from Africa, So these people often have triple negative breast cancer, which is the worst form of breast cancer. So breast cancer cells, they express 3 receptors. If all the 3 receptors are missing, those are called triple negative breast cancer. It's very hard to treat that, and it is the most aggressive cancer. So if someone has a cancer, you do the genetic testing and you say, oh, this is HER2 positive, this is HER2 negative, but this is triple negative. So then you know this is not going to respond to HER2 therapy because it's not even present here. But if HER2 is present, you give HER2 therapy. If HER2 is not present, you give something else, right? That is genetic testing, okay, or gene profiling, okay. Now, you can do it at a DNA level, but it is usually more advantageous if you go to the protein level. And it is a piece of cake nowadays to look at which proteins are expressed. So if you do all of these things, then you can pinpoint what is wrong with this cancer. So suppose somebody has a cancer. So what pathologists do, they do a biopsy, and then they take a tissue of that tumor and they do the testing. And then they can say, okay, this is what is wrong with this cancer. Okay, they cannot say precisely about everything because there could be 1,000 things wrong, but their kit only tests for 21 things. So they can tell confidently about those 21 things, not all the 1,000 things. But you can still be very precise. If you do a global proteomics or a global thing, you can find out a lot more information. That's where science is heading, to really find what's wrong with this particular patient. And that's what is precision medicine. You find exactly what is the culprit there, and then you treat that one. Okay, so all of you know that DNA makes RNA and RNA makes protein. So either you can test the DNA, but it is possible our DNA is perfectly good, but something goes wrong at this place. But at protein level, we know a lot of things go wrong in cancer. So if you can directly check the protein at proteomics, you do proteomics, you check all the protein levels and modification and everything, you can find out the culprit pretty easily. Okay, so that's what it is showing here. And if you do that, you know the exact things which are wrong, and you can treat it very specifically at that level. All right? So this is the end of the first lecture. All right, guys? Questions? Can I do we have enough time for questions, Venkatesh? Oh yeah, yes, go ahead.
Ma'am, like you told about the stage 2 cancer, where there is no metastasis, but there is in nodes there. So how it will be present in the nodes without metastasis, without going to the blood?
So what happens is that, so it metastasizes. So suppose somebody has cancer of the liver, right? So that liver cancer can go to the nodes, and I will show you pictures of the lymphatic system of the body. So you will see all of us, we have anywhere from 500 to 600 nodes, lymph nodes, right? So suppose somebody has liver cancer, so it's likely that it will go to the lymphs close to the liver, right? But it is still lodged there. It has not gone to the lungs and it has not made anything. But the problem is that once it goes to the lymphs, it is very easy to spread out, but it has not yet spread out. It is still confined to the lips. So I know of a person, she got mouth cancer a couple of years ago. She lives in Kolkata and she got mouth cancer. So, and she did not tell anyone. So her mouth was swollen and she let it go, not taking care of herself. And then somebody forced her to go to the doctor. By the time it was pretty spread out in the mouth. And she was in her early 40s. 2 years ago, 3 years ago, this thing happened. So she was rushed to Mumbai, okay? And one of the doctor who is Dhanvantari Award winner in Mumbai, he's a mouth specialist. So he removed all her lymph nodes in this area, all the lymph nodes. So it was pretty drastic surgery, okay? He even told us how many lymph nodes he removed, okay? But he saved her life. And it did not metastasize. So what I'm trying to say, once it goes to lymph nodes, it's not instantaneous metastasis. Okay, you still have some time, and if you remove those lymph nodes, you can prevent metastasis. Okay, but if it still metastasizes, people give chemotherapy or other systemic therapies to contain it, hopefully cure it. Okay, so lymph node does not necessarily means metastasis.
One more question. When we talked about the prostate cancer,
Yeah.
and there, like we have seen like phenotypic plasticity, that neuron like.
Yeah, neuroendocrine prostate cancer.
Like what induces the mutation in like phenotypic plasticity showing in a new.
See, that's what the whole world is trying to figure out. So there are many ways to do that. We have identified a lot of proteins that leads to neuroendocrine prostate cancer. So in our brain, for example, there is a protein called synaptophysin, which is a very famous neuronal protein. So these prostate cancer cells, so let me give you a little bit background on prostate cancer. So prostate cancer, prostate cells use testosterone, right? And so these prostate cells are addicted to androgen receptor signaling. So when normal prostate cell becomes prostate cancer, they upregulate androgen signaling, right? So the medicine we give is anti-androgen. Okay, so now we block androgen signaling, cancer disappears. So now when they become neuroendocrine, right, they don't need androgen. They don't care about androgen. They change their, what is phenotype? Look, how we look, right? They change their shape, they change their proteins, they change their signaling pathways. Now whether you give androgen, you don't give, it doesn't matter because they don't depend on androgen. So now they will upregulate synaptophysin things, and now they have a very different way of surviving by just completely changing themselves. You can keep on adding drugs, no effect. But they still are cancerous cells. These cells don't divide. Neurons, normal conditions, they don't divide. We are born with neurons. There is some neurogenesis, but not too much, right? But these are actual cancer cells. So when you, if you want to make neuroendocrine cells in the lab, you take prostate cancer cells, you slowly deprive them of androgen for very long, and you will see that they become neurite-like. We have done in our lab too. But the problem is that those cells don't divide like cancer cells. So something else is going on, that they are neurons-like, but they're still dividing like cancer cells. So that is, people think, is epigenetic modification. Something else is happening. We know a lot of players, but not all the players. So they upregulate MYC protein. MYC is something we'll discuss. MYC signaling upregulate, cancer pathways are upregulated.